going to be explaining transcription and translation here. I'm also going to be using this thing to like highlight some stuff that's important. So just at the very start of the process in the transcription stage, the RNA polymerase, this enzyme right here, it unravels the strand of DNA for the gene it needs to recreate. So, and it starts at the initiation site, and from there it creates an RNA strand. But instead of using the normal uracil that the DNA uses, it uses thymine instead. So the base pairs are, the complementary base pairs are AU and CG in the RNA, in the messenger RNA that is being created. So, and then, yeah, this copy is called messenger RNA, and once the polymerase reaches the end of the strand it's copying at the termination site, it breaks off, and the messenger RNA is able to leave the cell leave the nucleus through right here. And then it goes into the translation phase in the cytoplasm. So as it exits into there, it goes to meet up with ribosomes, which you can see right here, that have a large and small subunit. That's a really bad highlight right there. That's the small subunit. And then as you can see here, it's kind of gets stuck between the two, which is a good thing. So first we should talk about the triplet codes that the RNA has. These are called codons, you can see right here. And the first codon that enters into the ribosome is called a start codon, and it begins the polypeptide chain. Transfer RNA, or tRNA, will bring complementary codons, anticodons, along with a matching amino acid. And the tRNA enters the ribosome through the A site, and then at the P site, the polypeptide chain is created and bonded through peptide bonds. Sorry, I forgot the name for a second. And yeah, and then at the E site, the transfer RNA leaves the ribosome to return to the tRNA activating enzyme. I couldn't figure out that name. It's right here where they are designated a new amino acid, and they continue the work on the RNA. I think that's most of it for the tRNA. So yeah, the polypeptide chains right here created in the P site, that at the end of the messenger RNA, when it reaches the stop codon, it ends the polypeptide chain, and then you are left with an amino acid / a protein, which is the end goal. It's all a very streamlined process through and through to keep our body functioning.